It copies itself, which happens when? During the S phase of interphase.
Oh, shoot.
And basically how it works is there's a couple enzymes. One's called DNA helicase, and it comes and unzips the DNA, like that.
It's a protein, right?
It's a protein, yes. And the other is called DNA polymerase. And one by one, it takes free nucleotides and places them on the correct complementary base. So if this is a T, that'll be an A. If this is a C, that'll be a G.
So helicase breaks it and polymerase puts it back.
Helicase breaks it, polymerase puts on the new nucleotides and binds them together. Now, the special part of this, the part that's a little bit odd, is that if you look at this strand right here, this is where it gets hard, so pay good attention. If you look at this strand right here, it runs in the 3' to 5' direction. Do you see? Because the phosphate is the 5'. Well, the new strand has to be assembled in the 5' to 3' direction.
It's opposite, right?
Because they're opposite. In this strand, and anytime you start a new strand, it can be put on fine in that direction. And so as the helicase is unzipping, new nucleotides are being added right behind it. So the helicase unzips, new nucleotides being added. Helicase unzips, new nucleotides are added. And so this side is continuously copied, continuously replicated.
Which side?
This side right here.
Okay.
The DNA helix unzips and new nucleotides are put on. Unzips more, another nucleotide. Unzips more, another nucleotide. So right behind this, new nucleotides are being put on. We call that side that's continuously replicated the leading strand.
So the original strand is 3 to 5 and the leading strand is 5 to 3?
No, the leading strand is 3 to 5, and the new strand that's being put on is put on in the 5 to 3 direction.
So the leading strand is the strand that was already there?
Yes, that's the leading strand.
It's called the new strand, is what's put on.
Yes.
New strand. Very scientific.
I might have a name, I can't think of it. The other side is called the lagging strand.
Aww.
And if you notice, it's in the 5' to 3' direction. The new nucleotides have to be put on in the 5' to 3' direction. So they can't start up here and be put down this way. They have to start down here and be put on going upward. So what will happen is this, it'll unzip in another DNA polymerase. One DNA polymerase will be doing this side, and another DNA polymerase will come in and start doing this side, but we'll do it in the opposite direction. So you unzip, and then new nucleotides are put on starting down here, there.
Why is it? That's not. Would that be going from 3 to 5?
Hold on.
That's what you're trying to do.
There.
It's going to 5.
This one doesn't fit, but let's pretend. Oh,
You don't want to get that.
there.
Is the DNA
And there.
polymerase the thing?
So hold on. So this one's being put on in this way, and that's in the 5' to 3' direction. And this one's being put on going that way. And if you notice, that's also in the 5' to 3' direction. The new strand is always put on in the 5' to 3' direction. So the new strand is put on this way on this side, and that way on the other side. Hold on just a second. So what ends up happening is, as I unzip this thing, this strand is continuously being put on, and the other side is put on in fragments. There'll be a little fragment there that's put on, and I unzip some more, and another fragment here gets put on, and I unzip some more, and another fragment here gets put on.
So just top, bottom, bottom up?
Yes, bottom up, but this side has to be done in fragments, because it can't start until you unzip it far enough.
So the right side's going down and the left side's going up?
The right side's continuously going down. Wait, the left side.
It's going up. Or no. The
This
right
is left,
side's
right?
going down. The right side.
Just-
The right's going up.
This is right and this is left?
Yeah.
Are you saying
On the left.
you're
they're
saying
going down.
that this is the right side?
Yes. And they're going up.
The new nucleotides are put on going in that direction.
So they're going up.
In this model,
Yes.
but you understand this is a 3D model, and the whole thing can be turned around. And then you can't use left and right and up and down. You have to use 5' to 3' and 3' to 5'.
It's like your left atrium.
So when you say put on in fragments, it starts in the middle, goes up, and then goes
Starts in the middle, goes up, unzip some more, starts there, goes up, unzip some more, starts there, goes up.
So we should just say that it's done in fragments.
It's done in fragments, and these fragments have a name. They're called Okazaki fragments, after the guy who discovered them.
Can you spell that?
Okazaki fragments.
Okay.
So are there 2 leading and 2 lagging strands in each thing?
Nope. This side is the leading strand, that side is the lagging strand. The leading strand is copied continuously, the lagging strand is copied in Okazaki fragments.
So the leading strand will like finish before the lagging strand?
Yeah, but the process goes really fast, so they end up finishing about the same time.
Do you have any more videos? Or
Yes,
anything?
I'm going to show you videos. It shows it better than I can really show it with these models.
Where is the like DNA coming from that's getting attached to the leading?
These things, where are these from?
You know, they're just like
They're
floating.
either made by the Golgi apparatus, like assembled by the cells, or they're taken in by the cells from their external environment. Like, for instance, when you eat meat or something, those are cells that have DNA, and that's all broken down by your cells and made into your new DNA strands. Yes?
Are those amino acids or nucleotides?
These are all nucleotides. DNA is made of nucleotides.
What section is this?
Proteins are made of amino acids. Good?
Different chapter.
Yes?
Can you define Okazaki fragments again?
It's the fragments that are on the lagging strand of the DNA when it is replicating.
They're just like sections.
They're sections. So I have video footage showing this happening. And again, this guy's talking, he's using big words here, but you're going to have to be able to understand big worded discussion of biology.
We're not Navy Bio for nothing.
That's right.
DNA polymerase puts the nucleotides on, right?
Wait,
That's right.
Are those fragments leaving or getting put on?
They're getting put on.
What was it?
That's why they're called
DNA helix
Okazaki.
splits it. DNA helix splits it, and then
DNA replication will
Watch
begin
it.
at a specific area of the molecule called the origin of replication. The origin of replication denotes the area of active replication called the replication fork.
Fork in a road.
In order to understand how complex eukaryotic organisms replicate DNA, scientists first studied replication in prokaryotic models like E. coli. A number of enzymes are needed for replication to proceed once the replication fork is established. Helicase separates the strands of the double helix, and single-stranded binding proteins stabilize the newly single-stranded regions.
I didn't talk about
DNA gyrase is used to make sure the double-stranded areas outside of the replication fork do not supercoil.
I didn't talk about that either.
Do we need
That
to know
that
that?
cool
No, you
purple
don't need
thing
to
is
know that.
awesome.
That's when you.
Hold
What's
on.
that?
Let's try this one.
DNA replication begins at a specific point in the DNA molecule called the origin of replication site. Initially, the enzyme helicase unwinds and separates a portion of the DNA molecule, after which single-strand binding proteins react with and stabilize the separated single-stranded sections of the DNA molecule.
You don't have to know that.
The enzyme complex DNA polymerase engages the separated portion of the molecule and initiates the process of replication. DNA polymerase can only add new DNA nucleotides to a pre-existing chain of nucleotides. Therefore, replication begins as an enzyme called primase assembles an RNA primer at the origin of replication site.
I didn't talk about that
The
either.
RNA primer consists of a short sequence of RNA nucleotides, complementary to a small initial section of the DNA strand being prepared for replication. DNA polymerase is then able to add DNA nucleotides to the RNA primer and thus begin the process of constructing a new complementary strand of DNA. Later, the RNA primer is enzymatically removed and replaced with an appropriate sequence of DNA nucleotides. Because the two complementary strands of the DNA molecule are oriented in opposite directions, and the DNA polymerase can only accommodate replication in one direction, two different mechanisms for copying the strands of DNA are employed. One strand is replicated continuously toward the unwinding, separating portion of the original DNA molecule, while the other strand is replicated discontinuously in the opposite direction with the formation of a series of short DNA segments called Okazaki fragments. Each Okazaki fragment requires a separate RNA primer.
That's so cool.
As the Okazaki fragments are synthesized, the RNA primers are enzymatically replaced with the appropriate DNA nucleotides, and the individual Okazaki fragments are then bonded together into a continuous complementary strand.
So I wish they kind of ran that for longer, but you can see the top is just continuously being copied, and the bottom is copied in little fragments. You see?
Mm-hmm.
But it results in.
They both result. It results in two different strands. This bottom strand will fold up, will turn and form a helix just like this does. They should show that happening there.
Is the one that you're touching with your mouse, is that the lagging strand?
Yeah, this is the lagging strand and this is the leading strand.
Why do they have to be done differently?
Because they're oriented in different directions. This one is in the, like I drew up here, is in the 3' to 5' direction. This one is in the 5' to 3' direction. And the new strand can only be put on in the 5' to 3' direction. So on this side, the new strand can be constantly put on, because that's the right way. But on this side, the new strand has to go up. And you can't put on a new strand going up. You can't start from right here and do it going down as we unzip. You can't start at the top. You have to start at the bottom and go up. So you have to unzip a good ways and then start and go up. And unzip more and then start and go up. You see what I'm saying? Yes?
So I think I'm confused. Is the leading strand 5 to 3?
The leading strand is 3 to 5. The new strand has to go in the 5 to 3.
What is that RNA?
Where that's what I'm going to get to. I left that out on purpose because we hadn't talked about RNA. That's next.
When we're lured.
Okay?
Stop with the word.
Let's see it differently. They've actually, this process is a lot more complicated than I'm explaining it. And they put the complexities into these new videos, which are kind of cool, but we don't have to learn all this.
Once the replication fork is stable, DNA polymerase catalyzes the addition of new nucleotides to the growing daughter strand. Other proteins, such as beta clamps and the clamp loader, help hold the DNA polymerase in place on the DNA. Short sequences of RNA, called primers, have to be paired to the template strands by the enzyme primase, because DNA polymerase cannot begin to add nucleotides without a primer. Replication of both strands occurs at the same time, one using continuous synthesis and the other discontinuous. Continuous synthesis occurs on the 3' to 5' oriented parent strand, referred to as the leading strand. New nucleotides are added to the 3' end, moving continuously toward the expanding replication fork.
Dang it, they didn't show the lagging strand.
What's that cool purple thingy?
That thing?
Yeah.
Just another enzyme that's
That
involved.
is so cool.
Oh, here we go. Discontinuous synthesis.
Discontinuous synthesis occurs on the parent strand that is oriented 5' to 3', called the lagging strand, and is completed in segments called Okazaki fragments. Replication on this strand uses primase to add primers ahead of the 5' end of the lagging strand. DNA polymerase III then adds short sequences of nucleotides, the Okazaki fragments, to the primer, filling in the gap. As the helix is opened further, this process repeats until the entire strand is replicated. DNA polymerase I replaces the RNA primers with DNA nucleotides, and DNA ligase is used to ensure bonding between the fragments and the replaced nucleotides.
That video could have been made better if they had just gone a little bit longer here and showed you how it goes one after the other. But oh well.
What is a primer? What is that?
A primer is a little piece of RNA that goes on the DNA, and this actually will not work without these RNA primers. So let's talk about what RNA is.
It's ribonucleic acid.
Yeah, RNA is a little bit different from DNA. And I have RNA represented by these blue nucleotides. DNA is the white nucleotides. And so what we're going to do now, I've already showed you how to copy DNA to make two identical copies of the DNA. That happens every time the cell has to divide. You copy the DNA. Jordan, are you with me? Some of the hardest stuff for this next test right now.
Didn't you say the last test was the hardest test?
Probably. Yeah. But this is some of the hardest stuff for this test. Each test has its own hardest stuff. Okay, so
It's going to be an essay.
let me ask you this question. Do you know what the DNA is for? Why is it, what does it do? I mean, it can copy itself, but so what?
The code of life.
Sends messages. The code of life.
The Holy Grail.
These are decent answers.
It has all the things we need.
Has all the things we need. This stores information for making proteins. You know what a protein is? Remember that long chain of amino acids? Remember that? The polypeptide?
Primary, secondary, tertiary.
Remember that? There's a primary structure, and it folds into a secondary structure, and tertiary and quaternary. And these things are enzymes that can work for the cell. These are the cell's workers. So if you can make your workers, you can have life. Because the workers can do things, can put things together and build the cell, and all you need is the ability to make these. DNA is the code for making these. The code for making proteins. And what I'm going to show you in the next 2 days, the rest of today and tomorrow, is how that's done. How do we make a protein? How does this get put together? And what it does is the DNA code is taken to a ribosome. You remember the ribosomes?
Little dots.
The little dots? Well, this is a ribosome made larger. This is what it actually looks like. And I'm going to show you how the DNA code is taken to the ribosome, and then the ribosome puts together the protein.
So we're not talking
According to how the code tells it to be put together. Do you notice that this protein has a bunch of different colored amino acids? There's 20 different kinds of amino acids. And they got to be put in just the right order. For this protein, you got to go white, 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 pink, pink, white, blue, green, orange. And you can't have this orange out of place or that messes up the whole protein. We got to have each one in exactly the right place for it to form the right protein, to fold into the right protein. And all that is coded in the DNA. So I'm going to show you how we do it.
So basically like instructions.
It's instructions, yeah? Instructions for making proteins.
Which was? So
Yes.
Proteins, they do all this stuff. It was in 3.4, metabolism support, all that. This is all they
That's
do?
right.
Wow, we do a lot.
Metabolism, support, movement in the cell. It's all done by proteins.
Yeah.
Moving stuff through the membranes.
That's important.
Oh, yeah. So here's how we do it. First, the DNA is going to have to be copied. Because the DNA is the master plan, you see. And there's only one set of plans in each cell. It's in the nucleus. Imagine the nucleus is like a library with only one copy of each book. They're not going to let you just take one of the books out of the library. The only way to get information out of the library is to copy it. Imagine it's like the Library of Congress. You can't take any books out of there, but you can copy what's there and take your copy. Okay? Are you with me? So we got to copy this code first. So the first part of making a protein is called transcription. It's the copying of the code. And it's not copied into DNA, it's copied into RNA. And RNA are nucleotides that can copy the DNA and transport it and take it out of the nucleus. So let me draw a little membrane here. Here's the nuclear membrane. And we can leave the nucleus here and take the copy with us.
So the first step is they copies the DNA and then makes it RNA, takes it out of the nucleus.
That's correct.
Wait, can you repeat that?
Transcription is the copying of the DNA into an RNA copy.
This occurs during S phase.
So
No, this occurs during G1, during normal cell life. When during that time, cells are making proteins and using them in different ways. So this is going on all the time, basically. It even happens during mitosis.
So all RNAs is the copying of DNA code?
RNA is the molecule that we make the copy out of, which I'm going to show you how we do here.
It's like the Xerox machine.
Yes. How are we going to copy this DNA? Well, first we have to unzip the DNA a little bit.
helicase.
And it's not done with helicase this time. It's done with a different enzyme called RNA polymerase. RNA polymerase comes in. Let me move these nucleotides out of the way.
Check it
So
out.
why is this different?
It is. You're going to see. It's way different.
But why?
Because we don't want to make a DNA copy. We want to make an RNA
What's
copy.
the difference?
RNA can leave the nucleus. DNA can't. DNA is too big. It's double-stranded. It's too wide. It can't leave the nucleus.
That's annoying.
RNA is smaller and able to travel. Because it's got to get to the ribosomes,
Wait, it needs the whole.
which are in the cytoplasm, not in the nucleus.
Wait.
Yes.
So you need a whole DNA strand just for the information for one ribosome.
No, we don't need the whole DNA strand. And this DNA strand is real long.
Alright.
It goes off about 100 million nucleotides. So at this scale, it would extend all the way over the bridge. We only need a little part of it.
How many strands are in the nucleus?
46. Each strand is a chromosome.
How many feet are they?
Good question.
How long are they? You add
Well, they're all balled up in the little tiny nucleus of the cell.
46
stretch them out.
I don't know. It's a good question.
46 strands of DNA in a nucleus.
Each nucleus has 46 strands like this that are real long. Those are your 46 chromosomes. Right? The DNA balls up into a chromosome when we're about to divide the cell.
Don't like prokaryotes only have one strand?
Prokaryotes only have one chromosome, and it's in a loop. Okay, so here we go. The RNA polymerase comes in, and it opens up the DNA.
How is that attached?
I don't know. It's actually the
That's
RNA.
so cool.
Oh yeah, I've attached all these together with a string. And the RNA polymerase, one by one, will add RNA nucleotides to the DNA strand.
So it's
Click.
split up and attaches?
Yeah, it opens and attaches. Click. Click.
They're called RNA
So it's
nucleotides.
adding
They're RNA nucleotides. Click.
What is it added to?
The DNA strand, this is the DNA strand, and this is the RNA strand that's being built. Now it only attaches RNA nucleotides to one side. It doesn't attach to both sides. Because the RNA is a single-stranded molecule, it's not double-stranded.
Does it need, how much of the strand does it need? Does it need the whole thing?
It needs about, most proteins are about 900 nucleotides long.
Does it always copy the leading strand?
Yes, it always copies the leading strand.
Can it like copy from
Although you don't use the terms leading strand and lagging strand when you're copying, when you're doing transcription. That's only used when you're replicating the DNA.
What happens to it?
This side's just going to hang out there for a minute.
Enjoy.
Cole, what were you going to ask?
I mean, like, can it copy from like the middle of the
Yeah,
strand?
it's like 5.
Yes, it actually, this strand is real long.
Okay, so
And it just opens up the middle of it.
Okay.
It's like you had a real long zipper and you just kind of tore it open in the middle.
That's like a perfect way for me to think about it.
There you go. That's because I'm such a good teacher.
Obviously.
Thank you. Yes?
Rose, does it have to choose the middle, or it could be any part along it?
It chooses a specific part. As a matter of fact, the RNA polymerase starts at a location. This continues upward, and this continues downward. And the RNA polymerase locks on at a location called the promoter. There's a promoter, which is where the RNA polymerase comes in and attaches. And then at the bottom, there's a terminator where the RNA detaches. So it attaches at the promoter, opens up the DNA, one by one, attaching these little RNA nucleotides. Click, click, attaches them on. Click, click, and click. And then it'll keep doing that all the way down until it reaches the terminator, and then it falls off. And so it's made a long single strand of RNA. And we call that the messenger RNA, or mRNA. Yes, Elena?
Like in the book, like at the promoter, it starts like, I don't know how to explain it. Like it's like off to the side.
The promoter's off to the side?
No, the promoter's on the thing. But like the RNA is like, it comes off out from the side and comes in.
It goes like that?
Yeah, kind of.
Hold on, let me see. Is the picture...
It's on page 225. Yeah,
What
that's
we
it.
looked at yesterday.
Like down, like the bottom is the promoter down there.
Yeah.
It comes out.
Yeah. Well, the RNA polymerase stays on, and when the RNA polymerase gets down here, it falls off.
Okay.
You see?
What is the
So-
Oh, I'm sorry. They got it upside down from the way I'm doing it. The promoter is where it starts. So the RNA polymerase started down here and is working its way up, and it'll fall off at the terminator.
Okay.
So I'm doing mine down, and it's doing it up.
What is the promoter and terminator?
The promoter is where the RNA polymerase attaches.
It's just like a spot.
Yeah, it's a spot on the DNA. It's a certain sequence that the RNA polymerase looks for. AATTCGAA, and then it says, oh, I'll attach there. And then it goes down and does its transcription, and then it falls off at the terminator. Yes?
I don't get mRNA.
Okay. So we have attached these RNA nucleotides and made our little RNA sequence.
Yeah.
And this whole thing will then detach. And they're all bonded together, you see. And make its way out. We call this mRNA.
That piece that's leaving.
This piece of RNA that we've assembled from the DNA template is what we call this. This is a template strand.
Why did it attach in the first place? It was just really...
Because it has now copied the code. For instance, if this code was TCAAG, then this is a copy of it, because what attaches to T? A attaches to T. And what attaches to C? G attaches to C. So we have copied the strand here. You see? It's like a reverse copy. It's like a photographic negative. We have taken the order of these bases and copied them into the order of these bases. And they're in the correct order. All bonded together.
And those RNA.
They're RNA nucleotides.
So it's not like changing what they are, it's just like putting them in the right order.
Putting them in the right order.
What's mRNA?
mRNA is this.
The whole thing?
Yeah, we call that mRNA. Messenger RNA. It's taking a message and it's bringing it to the ribosome.
After the beat.
Boring. No, the Golgi apparatus is, we assemble the proteins first, and then they go off to the Golgi to be for further modifications.
The R?
No.
So where's the uracil?
Hold on. Okay, so RNA is different from DNA in a couple of ways. RNA is single-stranded. DNA is double-stranded. In this part, after it's done, we'll just go back together. So all we did was we took this apart, copied it, and then put it back together. Caitlin, you okay? Mm-hmm. Okay. So we have maintained our original copy. The RNA here is single-stranded, not double-stranded. It also has a difference to it. The sugar in an RNA nucleotide is not called deoxyribose. It's called ribose. So that's why it's called RNA and not DNA. DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid because the sugar is ribose here, where in DNA it was deoxyribose. It's a different sugar. Different sugar, different name. And there's one other big difference. The other big difference is in DNA, what is A normally attached to? T, thymine. But RNA doesn't have thymine. RNA has uracil, U, instead of thymine. So anytime there's an A on the DNA, you will attach to it a U on the RNA.
It's the gold spot.
That's annoying.
Yeah, it is annoying. And I wish it wasn't so, but that's the way life works. Uracil is the name of the base that attaches to adenine in RNA. In DNA, it was A bonds with T. In RNA, it's A bonds with U.
Why does the RNA break off? Like, what makes it break off?
This enzyme, after it puts it together, allows it to fall off. So it can leave the nucleus and go to the ribosome. The ribosome is going to read these instructions. This thing's going to cruise on out of the nucleus, and I'll talk about, there's actually some stuff done to it before it leaves the nucleus. But it cruises out of the nucleus and goes to the ribosome, and then the ribosome reads this code and assembles the protein. So you see, we had to get the code out of the nucleus. We can't take the DNA out of the nucleus because the DNA is too valuable. It's our only copy of the information. And if it gets out here, it's not protected by the nuclear membrane. So the purpose of the nuclear membrane is to keep the DNA here under protection so we can conserve our code, not have too many mutations and such. And whenever we need the code, we just copy it and bring it out. Cells that evolved a nucleus to contain the DNA information survived longer because the DNA wasn't damaged. Evolution. Took 2 billion years for a nucleus to evolve, but finally did. What do you think? All right, go away.
So it's AUG.
You've learned too much.